Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, let's unpack this. Imagine organisms so incredibly ancient, I mean really ancient, and so seemingly simple that for the longest time we scientists thought, they were just loners.
Right. Existing as single cells.
Exactly, just content to exist as single, isolated cells. But what if, and get this, a gentle, everyday squeeze, just a bit of physical pressure, could suddenly unlock this hidden potential, making them form complex tissue-like structures? This is where it gets truly fascinating, I think.
Absolutely.
Today, we're embarking on a deep dive into a groundbreaking new scientific paper. It's fundamentally challenging our very understanding of life's evolution.
It really is.
We'll be focusing on the astonishing details of how simple physical pressure might have acted as a primal trigger for multicellularity in, well, one of the oldest forms of life on Earth, Archaea.
Yeah.
Our mission in this deep dive is to really explore this incredible discovery and what it means for the vast, unfolding story of life on our planet and maybe even beyond.
It's genuinely a monumental discovery because, historically, Archaea have occupied this really peculiar and often misunderstood spot on the tree of life.
Right, they're kind of the mysterious ones.
We are. So just a quick primer for everyone. Archaea are one of the three fundamental domains of life.
Yeah.
You've got bacteria, eukaryotic, that's us, animals, plants, fungi, and then archaea.
Okay.
Now, visually, they often get lumped in with bacteria. They're both single-celled, microscopic.
Looks similar under a basic microscope, maybe.
Exactly. But biochemically, genetically, they are profoundly distinct. They occupy their own unique, ancient branch, and they're famous for their incredible resilience, right? Thriving in some of the most extreme environments you can think of.
Like volcanic hot springs?
Precisely. Superheated volcanic springs, incredibly salty lakes where almost nothing else survives, even the crushing pressures of deep-sea hydrothermal vents, they're tough.
Wow.
So for decades, the established scientific view was that Archaea were, well, almost exclusively solitary single-celled organisms. We kind of saw them as evolutionary holdouts.
Like they missed the memo on getting complex.
Sort of, yeah. Like organisms that had involved the sophisticated developmental programs that let more complex life, like us, differentiate and form intricate tissues and organs.
Okay.
And while we've seen multicellularity all over the place in eukaryotes, from our own bodies to giant kelp forests, and even sometimes in bacteria forming simple clumps or biofilm.
Right, biofilms, yeah.
We had never really seen anything like coordinated tissue-like organization in Archaea, not in a stable, repeatable way. So this left a pretty big conceptual gap in our evolutionary story.
Like, how did complex life really start if these ancient guys seem to lack that basic ability?
Exactly. How did complex multicellularity truly begin? This new research just steps right into that gap, and it's ready to bridge it in a really surprising way.
And bridge as they did, with a discovery that is, frankly, kind of jaw-dropping in its simplicity and its implications.
It really is.
So the absolute star of our story here is this fascinating organism called Haloferax volcanii, a salt lover. A salt lover, exactly. As the name suggests, it thrives in highly saline environments. Think salt ponds, maybe the Dead Sea. Normally, it's quite mobile, swimming around freely as a single cell.
Just doing its own thing.
Doing its own thing. Now, here's the completely unexpected trigger. The researchers found that when these Haloferax volcanii cells were simply subjected to a gentle physical pressure,
Just a squeeze,
specifically by being squeezed ever so lightly between two agar surfaces, that standard jelly-like stuff in labs, they completely transformed their behavior.
It's amazing.
It was a true aha moment, wasn't it? The kind that really shakes up scientific thinking. These single cells, which normally live totally independently, just abandoned their solitary life.
Yeah.
Instead, they started organizing themselves into incredibly ordered sheets. And what's truly striking is that these sheets looked remarkably like epithelial layers.
Which are fundamental for us.
Right. For animals, they're the foundational building blocks of our tissues. It's like, imagine you have a loose pile of arbels, right? And you just apply a gentle pressure, and suddenly, without any complex instructions, those marbles perfectly arrange themselves into a beautifully tiled floor.
That's a great analogy.
It's just mind-blowing to think that such a subtle physical cue can trigger such a profound, complex biological response. Makes you wonder what else is hiding in plain sight.
And what's truly captivating about this whole project is the meticulous way they pieced it together. Like detectives, really. How did this transformation actually happen? Moving from just a casual observation to a fully worked out mechanism.
So where did they start? What was the first hint?
Well, the journey actually began with a subtle clue. The team had been growing Haloferax volcanii, and they noticed something a bit peculiar. Their biofilms, those communities of cells that stick together, seemed to become unusually thick, and specifically right at the edges of the agar pads they were growing on.
Where they might be getting squished a bit.
Exactly. That little observation hinted that maybe some form of compression or physical constraint might be the cue.
Okay.
So this subtle thing, almost an experimental artifact you might ignore.
Right.
It sparked this bold, really audacious hypothesis. Could mechanosensation, just the cell's ability to sense and respond to physical forces like pressure or stretch, could that be enough on its own to make archaea reorganize themselves into these complex structures?
Without needing a whole bunch of new genes or complicated internal parts.
Precisely. Without needing entirely new genes or complex internal machinery like our cytoskeleton, if that were true, it would reveal a kind of primordial developmental logic, a really ancient, fundamental way life organizes itself.
Predating the complex stuff in eukaryotes.
Exactly.
Right.
Predating things like our intricate cytoskeleton, which is so crucial for our cell shape and division. That was the big question they set out to tackle.
So they had this incredible idea, this hunch, that pressure was the key.
Yeah.
How did they actually go about proving it? What tools did they use on such a tiny ancient organism?
That's where the really ingenious experimental design comes in. To precisely watch this cellular choreography under controlled conditions, they developed these custom-built uniaxial compression chambers.
Okay, fancy name. What does that mean practically?
You can basically picture them as tiny, highly specialized vices. They let the researchers apply really precise, very gentle forces. We were talking kilopascal level forces.
Which isn't much pressure at all, right?
No, it's very mild, like the pressure you might feel from just a few drops of water. So applying these gentle forces directly onto the archaeal cells, and then to actually see what was happening in real time, they used this cutting-edge technique called time-lapse lattice light sheet microscopy.
Now that sounds like a mouthful, but it's a powerful technique, right?
Okay.
For our listeners, what does that actually let them see that a regular microscope wouldn't?
It really is powerful without getting too bogged down in the optics. Imagine shining an incredibly thin, almost perfectly flat sheet of laser light through your sample. This lets them illuminate just a very narrow plane of cells at a time. So they can capture incredibly detailed 3D images of what the cells are doing, second by second, without blasting them with too much light and damaging them.
So it's like getting a super high-def, real-time movie of cell behavior.
Exactly. It allowed them to track individual cells as they responded to that gentle pressure.
And what did they see in that movie? What was the choreography?
Well, what they captured was truly remarkable. Under this gentle compression, the cells didn't just immediately divide like you might expect.
No.
Instead, they went through this peculiar phase of just enlargement. They grew significantly bigger without splitting, just kept getting larger until they hit a critical size.
Okay.
And at that point, it seems changes in their cell membrane tension triggered a process called cellularization.
Cellularization, what does that involve?
For many organisms, it means forming individual cells within what might have started as a larger shared structure, maybe with multiple nuclei. But crucially, in Haloferax, this process turned out to be tubulin independent.
You mentioned that before. That's a key distinction, right? Because it means they found a different pathway for this kind of organization than what we typically see in complex life.
Precisely.
Why is the tubulin-independent part so significant for us listeners?
Because in most eukaryotes, that's us, plants, fungi, animals, the protein tubulin is absolutely fundamental. It's a main building block of our cytoskeleton.
This cell's internal scaffolding.
Exactly. That internal scaffolding is crucial for cell division, maintaining cell shape, organizing everything inside. The fact that Haloferax volcanii achieves this impressive cellularization and tissue formation without relying on tubulin, well, it means they've tapped into a much more ancient, perhaps even universal mechanism.
Something that came before the complex machinery we eukaryotes evolved?
That's the idea. It suggests a simpler, maybe more primordial way that cells can come together and form structures.
Wow. Okay, so they cellularize without tubulin. What happens next?
Following this cellularization, the tissue junction elasticity, basically, how stretchy and connected these new cellular sheets were, became surprisingly similar to actual animal tissues.
Like our skin.
Yeah, think about how skin stretches but holds together. These archaeal sheets showed a similar kind of mechanical property. And even more intriguing, within these organized sheets, two distinct cell types actually emerged.
Different types of cells from just pressure.
Yeah, peripheral or pur cells, which form the outer edges, and central scutoid or skew cells.
Scutoid cells, that sounds like something out of a geometry textbook. What are those, and why is their appearance significant here?
Good question. Scutoid cells are actually a relatively recently identified geometric shape. Nature uses them for incredibly efficient tacking of epithelial layers in 3 dimensions.
Like in our skin or organ linings?
Exactly. How cells fit together perfectly in curved tissues. Their appearance here is super significant because it shows these archaea, under pressure, aren't just clumping randomly. No, they're adopting a very specific optimized cell shape known to be crucial for building stable functional tissues.
That's incredible planning, almost.
It seems like it. And both cell types, the peripheral and the scutoid, also showed unique patterns of actin organization and something called protein glycosylation polarity.
Okay, protein glycosylation polarity. Break that down for us. What does that tell us about their organization?
Right, so think of polarity as cells developing a clear top and bottom, or maybe a front and back, for specific jobs. It's fundamental for organizing functional tissues.
Makes sense.
And glycosylation is just the attachment of specific sugar molecules to proteins. So protein glycosylation polarity means these cells aren't just structurally oriented, but specific sugar molecules are being stuck onto their proteins in distinct ways at specific locations.
Like little flags.
Kind of like little flags, yeah, guiding their orientation and function. It's a clear hallmark of highly organized tissues, even in these seemingly simple guys. It shows a sophisticated level of internal organization triggered by pressure.
So they'd seen the physical transformation, the unique cellularization, even different cell types with specific orientations popping up. But to really understand how the pressure was pulling the strings, what molecular switches were being flipped.
But they had to go deeper, right?
We're-
Down to the
Exactly. That's where they brought in another heavy hitter, single-cell RNA sequencing. On a massive scale. They compared the gene activity of 20,000 of these compressed cells against 20,000 cells just swimming freely, the planktonic ones.
Wow, 20,000 each. That's a lot of data.
A huge amount.
Yeah.
And this allowed them to uncover what they termed a mechanoresponsive regulon.
A regulon.
Yeah, basically a specific group of genes that get activated or switched on specifically by that mechanical force.
The pressure turns these specific genes on. Correct. And this particular regulon was notably headed by a gene for a stretchy S-layer glycoprotein. The S-layer is that outer protective protein coat on archaeal cells, and also a 2-component signaling kinase.
A kinase, that's like a molecular switch, right?
Exactly like a switch. It receives signals from the environment, in this case pressure, and then activates other cellular responses, often by changing which genes get expressed.
Okay, so they're zeroing in on the pathway.
They are. And to understand the physical changes better, they also used atomic force microscopy. It's a technique that can literally feel the surface of cells at the nanoscale.
Feel them. How?
It uses an incredibly tiny probe to tap gently on the cell surface, measuring how much force it takes to deform it. And this revealed a dramatic 5-fold stiffening of the archaeal cell surface when it was under compression.
5 times stiffer just from being squeezed.
Yep. But here's the truly astonishing part, the real scientific mic drop moment, I think.
Okay, I'm ready.
This stiffening happened alongside the emergence of specific apical basal polarity proteins.
Right, the top-bottom defining proteins you mentioned.
Exactly. Proteins essential for establishing those distinct top and bottom ends in cells, which is absolutely foundational for building layered tissues like our skin or gut lining. And the real kicker, these archaeal proteins are homologous to eukaryotic para.
Wait, homologous to eukaryotic para. Can you explain what that means for us? That sounds like a direct, really shocking parallel to animal development.
It absolutely is. Homologous means they're structurally and functionally similar, strongly suggesting they share a common evolutionary origin way back.
Wow.
Paraproteins in eukaryotes, in us, are critical for establishing cell polarity, for proper cell division, for tissue organization during our own embryonic development. finding that archaea possess and activate proteins so similar to our own foundational developmental machinery, just under mechanical stress. It's profound.
It suggests an incredibly ancient shared blueprint for building tissues.
That's exactly what it suggests. A blueprint that predates the split between eukaryotes and archaea, pushing the origins of these fundamental developmental mechanisms much, much further back in time than we ever imagined. It's like finding the same basic Lego brick design in incredibly ancient ruins and also in a modern skyscraper.
Incredible. Just incredible. Okay, so they found the genes, the proteins, they saw the structural changes, but then they had to definitively prove that the force causes these genes to switch on and drive the whole transformation. How did they close that loop, make that final connection?
Right, the cause and effect. To confirm this force-to-gene circuit, that direct pathway from mechanical input to genetic output, they use these advanced gene knockout microfluidic arrays.
Okay, sounds complex again. Simpler terms.
Basically, they use tiny devices to precisely handle the cells, and they selectively deleted or knocked out that key two-component signaling kinase gene they'd identified earlier.
Yeah.
The one heading the mechanoresponsive regolith.
The switch.
The switch. And the proof was just undeniable. Deleting this specific kinase completely abolished the sheet formation. The cells got squeezed, but nothing happened.
Wow. So that kinase is the crucial link.
It is. It directly confirmed that clear pathway.
Oh.
A mechanical force flips the kinase switch, the kinase changes gene expression, and that drives the multicellular transformation. It's a complete, albeit minimalist, developmental program, all kicked off by simple compression.
And there was one more piece, right? Something about lineage.
Yes, finally, using computational lineage tracing, they basically tracked the descendants of individual cells over time as the sheets formed.
Following the family tree.
Exactly. And this showed that the sheets expand clonally, meaning they grow from a single ancestral cell dividing over and over. And this clonal expansion created something that looks strikingly like an early form of germ layer.
Germ layers, like in embryos.
Precisely. Germ layers are those foundational cell layers that form in early animal embryos and give rise to all the different tissues and organs.
Wow.
The fact that Haloferax forms something analogous under pressure is just astounding.
Wow. So putting it all together, these experiments depict this minimalist but complete developmental program, from sensing a simple cue to cellularization to forming distinct cell types with polarity, even expanding clonally like an early embryo layer. All initiated by just a gentle squeeze.
That's the story.
It's like finding a hidden switch that unlocks incredibly complex architecture with just a simple push. It really makes you rethink everything you thought you knew about these ancient, supposedly simple organisms, doesn't it?
Absolutely.
Yeah.
This research profoundly reshapes our understanding, not just of archaea, but of the tree of life and the very definition of multicellularity itself. Prior studies hinting at multicellularity in archaea were mostly anecdotal.
Right, hard to replicate, maybe.
Often, yeah. Lacking detailed mechanisms, hard to replicate consistently. But this paper, by showing a reversible compression-triggered transition that faithfully recapitulates key features like polarity, cell adhesion, clonal expansion, it fundamentally reframes multicellularity.
How so?
It reframes it not as a one-off evolutionary leap, but as a tunable physical biological state.
A tunable physical biological state. That's a huge conceptual shift. What does that really imply for how we view evolution?
It's transformative, really. It suggests the origins of complex life might not have been just a series of incredibly rare, super lucky genetic mutations happening in isolation.
Yeah.
Instead, it paints this picture where multicellularity could be activated by ancient, maybe pre-existing on switches for cooperation and organization. Switches that are readily triggered by common environmental forces, like physical pressure.
So life might be inherently more flexible and more responsive to its physical world than we thought.
Exactly. With a deeper shared blueprint for complexity, potentially. And this fundamentally challenges that long-held assumption that sophisticated development, like forming complex tissues, absolutely require the elaborate internal structures of eukaryotic cytoskeletons,
Which we thought were essential.
which were thought to be a prerequisite for that kind of organization. This finding strongly suggests mechanical forces could have been universal catalysts for early tissue evolution across all domains of life, not just eukaryotes.
Wow. So maybe similar things are happening in bacteria too. We just haven't found them yet.
It certainly begs the question, doesn't it? Are similar mechanisms waiting to be uncovered in other branches of life? And what's also incredibly inspiring here is the sheer interdisciplinary nature of this work. It wasn't just one lab.
Right. You mentioned several institutions.
Yeah, it bridged mechanobiology experts at Brandeis University with archaeogenetic specialists at the Weizmann Institute in Israel. They fabricated these custom microfluidic chips in a shared nanofabrication facility. They even did high-resolution cryo-electron tomography on the structures at EMBL Heidelberg in Germany.
That's a massive undertaking, coordinating all those different specialties and locations. It really highlights the global collaborative nature of cutting-edge science today.
It truly does. Apparently, they even had weekly Zoom data jams to synchronize everything, working across 3 different time zones, just constantly sharing and integrating results.
Incredible dedication.
Yeah, and it's always important to give a nod to the brilliant minds behind these breakthroughs. Folks like graduate student Theopi Rados, who designed those compression rigs and did the amazing microscopy, postdoc Shani Levy, who tackled the complex single-cell transcriptomics, their PI, Dr. Yoav Soen, who provided that crucial evolutionary framework, and their collaborator, Dr. Tanmay Varat, who was key for the structural validation with tomography. A real testament to teamwork.
Absolutely. Well, this deep dive into Archaea certainly reminds us just how much more there is to discover, especially in the microscopic world, and how seemingly simple physical cues can unlock incredibly complex biological phenomena. It makes you wonder, if mechanical forces can trigger such profound changes in these ancient life forms, what other hidden developmental potentials were just waiting to be uncovered, both out there in nature and maybe even in our own understanding of biology?
It opens up so many questions.
It really does. And this raises an important question for you, our listener. If such a basic physical cue can unlock multicellularity in these ancient organisms, what does that imply about how simple or maybe how complex the origins of life on Earth or maybe even on other planets might have been?
It's a fascinating thought.
The researchers themselves suggest that future work needs to figure out where exactly these kinds of transitions might occur in natural settings, like the subtle pressures inside sediment pores or maybe within dense microbial communities.
Yeah, finding it in the wild.
And also, whether similar mechanogenetic circuits, these force-to-gene pathways, exist in other domains of life, like bacteria. The story of life, it seems, just keeps getting more fascinating, constantly surprising us with its ingenious simplicity sometimes.
It never fails to surprise.